From the JAMA Network, this is JAMA Ophthalmology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Ophthalmology.
Hello and welcome to our listeners around the world. You're listening to JAMA Ophthalmology Author Interviews. Thank you all for joining us. I'm Neil Bressler, Editor-in-Chief of JAMA Ophthalmology, and today we're going to look at an interesting investigation into mTOR inhibition as a possible therapeutic target to prevent proliferative vitreoretinopathy, which we will probably abbreviate as PVR. And joining me is the corresponding author of this manuscript, reporting the results, Dr. Ehsan Rahimy. Dr. Rahimy is the Director of Vitreoretinal Diseases at the Palo Alto Medical Foundation. Ehsan, thank you so much for joining us.
Neil, it's great to be back on the podcast. Very excited to talk about this topic and looking forward to our conversation.
Fantastic. Well, let's just start with what is mTOR inhibition? This is not something that we talk about in ophthalmology all the time.
Yeah, you're right. And, when this was all starting, it just brought back memories to being in medical school and learning about these pathways. But mTOR stands for Mammalian Targeted Rapamycin. This is a very important serine threonine protein kinase. And the way to think of it essentially is it's sort of like a master switch or a regulator for cellular anabolic and catabolic processes. So essentially it's helping to regulate the rate of cellular growth and proliferation, which if you think about it, kind of makes sense in the context of what PVR is when it's developing in the eye and causing recurrent retinal detachments. So there have been various basic science papers that have potentially implicated this pathway as being a therapeutic target of interest when looking for pharmacologic therapy against PVR.
So before we get into the investigation you did, give me an idea of who's on mTOR inhibitors and how common are these? Is this very frequent? Is it really 1 in a million people? Who's taking these in the US?
Great question. These medications are actually quite common if you take the time to ask your patients and review over their medication reconciliation. Currently, there's 3 approved mTOR inhibitors in the United States by the FDA. There's sirolimus, which is probably the most commonly known agent, everolimus, and temsirolimus. They're approved for a variety of indications, including numerous types of cancers, blood dyscrasias, vascular malformations. They're often used for some more of these genetic conditions like tuberous sclerosis. And really the most common reason why they are prescribed is to prevent transplant rejection after an organ transplant. And that's the most common reason why I see patients on these. I'm kind of admittedly in a bubble in the Bay Area. And one of the reasons I keep seeing these medications used more and more often is there is basic science, animal-based literature that being on low doses of rapamycin, which is sirolimus, potentially helps with improving longevity and long-term outcomes and surviving longer. So we have a lot of people in Silicon Valley that are taking low doses of these medications as well.
So I understand you and your colleagues have a lot of experience searching through very large databases to look for associations, and we'll come to that in a minute. But in general, were you specifically looking for mTOR inhibitors and their association with PVR, or were you looking at mTOR inhibitors to say, is it associated with any retinal disease? Or were you looking at PVR and saying, are there any drugs that might be associated with preventing this? So what came first, the chicken or the egg? How did you design this? Was it a fishing expedition? Was it pre-specified very specifically that we're going to look at whether mTOR inhibitors have any relationship with reducing the development of PVR?
It's a great question. I think the nidus for this is more of the former and not just mTOR going in. Our group has a specific interest in looking at immunosuppression and the incidence of PVR. So just at a very high level, one thing that has me excited about this topic is if you think historically, we've seen the rise and the fall, and now again, I think the rise of enthusiasm over pharmacologic intervention for prevention and treatment of PVR. We've all seen a myriad of studies done over the decades looking at various things, steroids, other anti-metabolites like 5-FU. People tried anti-VEGFs with kind of mixed to disappointing results. And if anything, over time, our understanding of the PVR process has only grown, but also the advent of better targeted therapies, immunotherapies, immunosuppressive therapies, where we should hopefully be able to solve this problem at some point down the road. And no matter who your horse is in this race, we identified mTOR. A lot of other colleagues are looking at other pathways. There's investigations into Rho kinase and metformin. I think a lot of this enthusiasm has come back because of some of the promising results we saw with methotrexate in the GUARD trial. So that's where we were coming in, is kind of looking at broad spectrum immunosuppression and onset of PVR. And within that, looking at specific classes of medications to see, at least based again off of previously published basic science literature, should certain classes maybe have more of a protective benefit than others. And out of all of them, we saw the most promising signal from mTOR pathway.
So give me an idea of how many immunosuppressives you were searching through. Was this 1 out of 5, 1 out of 500? What was the search involving?
Class-wise, we had pretty targeted spec. We were looking at mTOR, we looked at methotrexate. That's a separate investigation. Obviously, there's been a lot of enthusiasm about methotrexate, be it administered intravitreal or for patients on it systemically. We were also interested in the TNF alpha blockers. As I'm sure you're familiar in the readers, there has been some promising results. I believe it was called the FIXER trial, looking at adjunct intravitreal administration of anti-TNF after complicated RD repair, which showed some, looked like promising results in earlier phase trials. And we started with those targeted groups.
And was this the biggest one that you found? Were the others close behind, or this is really the one of those classes that you found really stood out?
We saw trends across the board, which I took that as being promising. But I think there's other reasons why, yes, we saw the most promising results with mTOR. But if I'm actually thinking down the line, assuming we had something that's efficacious in the prevention and treatment of PVR, like how would we design a perfect drug to be administered? And I think ideally we want something that's well tolerated, pretty safe, orally administered, something we can start perioperatively around the time of surgery, something that's rapid onset of action, reaches therapeutic levels fairly quickly to help protect you during that window of risk, and then that you can kind of discontinue it. And I think a lot of these characteristics, mTOR inhibitors actually nicely check off many of these boxes. They are pretty safe. There's obviously some degree of side effects. They have rapid onset of action. They do reach therapeutic levels fairly quickly within the first week or so of administration. So if we imagine a future, if they did happen to work, I think it's just the means of delivery and use and how it would be used is somewhat optimal for what we need for surgery.
That is really helpful for framing the background to this. Now, before we get to how you actually did the investigation, maybe just for the general ophthalmology audience, you can remind us what's the current understanding of what PVR is? How common is this after retinal detachment surgery? What's the current management before you did this investigation? Just give us the 1-minute summary on PVR.
Absolutely. Well, the very top liner is PVR is the bane of our existence surgically. It's very frustrating when we all deal with it. It still happens. We haven't found a great way to eradicate it, prevent it, or completely treat it. We know that it's the most common cause of failure of a primary retinal detachment repair. And depending on what studies you look at, it occurs anywhere from 5 to 10% of the time after initial RD repair. And the process PVR at a cellular level is characterized by abnormal glial cell proliferation, migration, and then eventually there's contracture of the scar tissue and membranes, which induces a retinal detachment after initial repair.
So we definitely want to try and treat that and get rid of our main cause of failure. So maybe now you can walk me through, how did you investigate the association of these mTOR inhibitors with the development of PVR?
This is always the fun part for me. And as you mentioned, we do a lot of this work in these large databases. It's aggregate, de-identified electronic health record data. And obviously there's a lot of pros to this. There's a lot of cons to this. We can discuss that as well. But the particular network we're using is the Trinetics network. There's over 250 million patient lives captured in this, over 120 different institutions. So we're dealing with a very large pool of potential eligible patients. And I think the important thing is how we construct our cohorts to capture the right patient population we want to investigate, and then also to design a good synthetic cohort that is adequately matched to compare these outcomes to our actual investigative group. And so what we did in this particular study is we looked for patients who had an instance of a retinal detachment that underwent an initial repair. And this is where having knowledge of ICD-10 coding and CPT coding is critical if you do big data analyses. So for primary surgical RD repair, we included the code 67107, 67108. That's for primary buccal repair or a vitrectomy plus or minus a buccal, as well as including pneumatics as well too, which is 67110. Of note, just we excluded certain things too because we wanted to mitigate the potential for confounders here. We basically got rid of any pediatric detachments. As we all know, those just behave very, very differently from the adult population. So anybody under 18 years was excluded. Anybody with known diabetic retinopathy was excluded. That was important, again, because once you start getting into proliferative disease, the need for diabetic vitrectomy, this starts overlapping a lot with the use of the complex retinal detachment CPT code, which is 67113. So we excluded anybody with known retinopathy. We didn't want to have any chance that could kind of confound our findings. In line with that, we excluded certain medications. Metformin, for example, was excluded, which is of interest because this is a medication that is being investigated as potentially a PVR preventing or treating drug as well. And of note, metformin actually acts in one way as an indirect mTOR inhibitor as well too. So in particular, with how do we confirm mTOR uses, we wanted patients to have been on it for 3 months prior to their surgery date and up to a year after. And we had multiple checkpoints in place to make sure it was administered as a prescription in the record. We also nicely designed a positive control where we assessed for the side effects that are known to happen with mTOR use, stomatitis, dermatitis, hypertension. And we saw that compared to our control group, we saw a higher incidence of this in the mTOR group, somewhat confirming that it's being used. And the primary outcome we were looking at here was the incidence to get a redetachment, which again, this is where coding becomes complex. We're using as a proxy for PVR tractional retinal detachment, which is the ICD-10 code H33.4. Unfortunately, there's not a specific ICD code for PVR. That's one of our frustrations. Maybe in future iterations of ICD, we'll fix that. But until that time comes, we use H33.4, which is a nice surrogate. And we looked at the need for subsequent vitrectomy intervention as well as complex retinal detachment repair.
So this is really helpful. So now we're going to get into the meat of the matter, and that is you found that there was a reduced risk of this redetachment rate based on how you all defined it. Can you give me an idea of the magnitude of that reduced risk if people were on mTOR inhibitors? What was the relative reduction in the risk? And then we want to get into the absolute reduction.
And if you're looking at the numbers that we included, once we applied all this inclusion-exclusion criteria, we had about 681 patients. And then the goal here was to propensity score match them to our synthetic control group, which drew from a pool of over 47,000 individuals. So we had a broad heterogeneous pool to nicely match and get individual controls with our mTOR group, which we did. So the reduction that we saw in terms of reduced risk of developing a tractional retinal detachment, needing subsequent vitrectomy, which is, again, the proxy for that was 67108 code, or complex RD repair, which is the CPT code that's supposed to be used for PVR detachment repair, was between 40 to 43% risk reduction out to a year, which in terms of significance was definitely statistically significant. If you can have that much of a reduction, that seems very promising and definitely warrants further investigation.
And give me an idea if somebody has 100 retinal detachment repairs that they do and their event rate of having PVR, we'll say, is 20% for whatever reason. How many fewer PVRs might they have based on what you found?
In this case, you're looking at potentially 5 to 7 less redetachments, which over a course of 100 surgeries, I take 1, 2, or 3 less. They're a headache for us. My heart always sinks when I see a patient coming in with a redetachment, and anything I can do to potentially mitigate that risk is welcome. Now, this gets into a conversation about number needed to treat to prevent an event. And I'd argue the most important thing on the flip side becomes, well, what is the cost of such intervention? What are the potential side effects and safety profile? And again, assuming we were in that future where mTOR worked, I think this is where these medications shine. They're not overly expensive. Generics are available for them. They're safe and well tolerated. So I think to that end, it would justify the use.
All very important points. So finally, I'm sure you'd agree, you talked about the future of treatment. Should this change our practice yet? And should our retina surgeons be prescribing these? Or is this just an association and it could actually be harmful? We better test it first in some sort of ideally randomized clinical trial design.
100%. Anytime any of our findings from our previous large data studies, I always overemphasize this is like the bird in the sky, 30,000 foot view from above. we're finding signals, we're finding interesting findings. I don't like to use these type of databases for hypothesis generating. I like to see that there's already a basis for something and that we're exploring something that is known, in this case, looking at agents that can potentially help mitigate PVR. I want to give a shout out to he was a resident, now he's a first-year fellow at Beaumont. Amr Al-Sudi did a fantastic job with this project. He is actually looking to carry this forward into more of a model that he's going to test in the lab, and we're hoping to potentially lead this into an IST. So it definitely needs to be followed up and researched further. With that being said, I think the thing that's most exciting about all this is, again, who cares what your horse is in the race? I think what's important is that the race is back on, and I think that's what's exciting. It feels like we went through a 5-10 year period where it almost felt like our field gave up on PVR, and I don't see any good reason why we should. Our diagnostic acumen is so great. Another one of the supportive papers for this was from a colleague of ours, Dr. Nathaniel Roybal and Vinu Mahajan. They did nice proteomics analyses where they took liquid biopsies from patients who had PVR, and they implicated the mTOR pathway and downstream regulators. So having such advances in science and medicine with more targeted therapies, we should be able to find a solution or something to help reduce the risk of this process. Because again, whether it's mTOR, whether it's Rho kinase, whether it's metformin, or some of the other various agents being investigated, I'm most excited about the fact that our field is again enthused about the possibility that maybe we can find a treatment for this.
Well, thank you so much to you, to Dr. Al-Sudi. I look forward to following his career as well. And really, we need these sort of treatments, so we're glad that you're pursuing it. I'm just very thankful you shared that with JAMA Ophthalmology, and we look forward to the next steps. So thank you, Ehsan, so much.
Neil, thank you so much for having me. It's been a great discussion.
I'm Neil Bressler, and I've been speaking with Dr. Ehsan Rahimy on a evaluation of the possibility that mTOR inhibitors may be a potential target for preventing proliferative vitreoretinopathy. You can find a link to the article in this episode's description. This episode was produced by Daniel Musisi at the JAMA Network. And to follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. Thanks, everyone, for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.